Hello, I'm Marvin Hicke, and this is my independent project, Cultivating Genes, Evolutionary Shifts in Neolithic Agricultural Adaptations. I worked on this with my project mentor, Danju. So, what was the Neolithic and how did it begin? So, this period, also called the Agricultural Revolution, marks a stage when populations around the world independently transitioned from nomadic hunter-gatherer societies to settled agricultural communities. As early as 10,000 BC, some of the first groups began practicing agriculture. This revolution marked a change in humans' hunter-gatherer diets to ones that depend on domesticated plants and animals. The change to an agricultural diet and an environment with new selective pressure set the ground for adaptations to these dietary changes, including adaptations to consumption of dairy and carbohydrates. This review investigates several key genetic changes that occurred during the change from a hunter-gatherer to an agricultural lifestyle. So before the plow. So before humans' diet shifted toward agricultural products, pre-Neolithic humans lived nomadically as hunter-gatherers. As nomadic hunter-gatherers, they depend on hunting, fishing, and gathering wild plants for sustenance. Due to their lifestyle of constant migration, nomadic peoples were not able to stay in one area for long before having to find more sources of food, precluding the development of more complex societies and technologies. Their technological advancements were limited to lighter and mobile technologies such as stone axe, hand axes, animal hides for warmth and shelter, as well as spears and arrows. The last ice age, known as the last glacial maximum, was a possible factor contributing to this period of limited progress, as humans had to migrate due to changing climate and the movement of game. The unpredictable climate during this period is a possible reason as to why Paleolithic humans were unable to access and domesticate grains as a food source. Around 10,000 BC, the Earth was emerging from the last glacial maximum, and the climate was becoming warmer and stabler. This climatic stability was pivotal, as it could have allowed communities to settle and advance their technologies. As the ice melted, lands became more accessible, which possibly enabled the cultivation of grains. The changes that came with this period saw genetic adaptations in response to a different diet and lifestyle. The shift from a hunter-gatherer diet to an agricultural one entailed a more stable food source, as there's possibly more predictability with such a diet, being able to control factors such as irrigation and fertility of soil. The Neolithic contained many genetic changes to populations all over the world for the adaptations toward an agricultural diet and lifestyle. Many of these changes still affect people today and can be essential to our daily lives. This paper aims to describe and explain some of the important genetic adaptations to the agricultural-based diet during the Neolithic. So, lactase persistence. Certain traits were under selection in response to selection pressures during the Neolithic environment. One of the most well-known and important of these genetic adaptations was the autosomal dominant trait lactase persistence. The emergence of the capacity to digest milk in some populations is an example of adaptation in response to niche construction. This is the process by which an organism alters its own or another species' local environment. In this case, taking advantage of the milk that some animals produce. Like other mammals, all humans are born with the ability to digest lactose, but by default, this trait is lost around the age of weaning and the lactase is downregulated during childhood. The people in some regions domesticated dairy animals such as cattle, goats, sheep, and camels. Alleles promoting lactase persistence independently evolved around Europe, East Africa, and the Arabian Peninsula within the last 7,000 years. Those who had LP could take advantage of drinking milk into adulthood. Lactose provided an additional source of energy for those who had evolved the phenotype of LP without the harmful side effects. Without LP, consuming lactose past the cessation of the production of lactase enzyme during early childhood results in uncomfortable to harmful side effects. So, evidently, LP promoted fitness in humans. Although the underlying mechanics of it remain unclear, however, there are some hypotheses to the value of fitness from LP. Dairy animals such as cattle produce in a lifetime as much as 400 to 600 kilograms of milk, and that milk is extremely high in protein and fats, making it more valuable long term than consuming the meat. In addition, in times of famine when agriculture fails, the dairy animals could still provide that energy, making carrying LP extremely valuable. Milk drinking could have also played a role in elevating IGF-1 levels, leading to fitness benefits such as early maturation and increased body size. Not only does milk provide energy, but it also is an important source of calcium, which promotes skeletal health. It also provides small quantities of vitamin D, which help the body absorb calcium, preventing osteomalacia, which weakens bones. Weaker bones would reduce fitness greatly in the labor-intensive Neolithic, which consisted of much plowing, irrigation, and terracing. This was especially helpful for European populations living in higher latitudes, as vitamin D is less available through skin synthesis at higher latitudes, and also because early farmers had diets that were deficient in the vitamin. LP was valuable to peoples and regions that held dairy animals by providing energy, especially in periods of famine, and also by providing calcium and vitamin D, which could have improved fitness through increased body size, earlier maturation, and stronger skeletal structure. So here are fads. Fatty acid desaturates, or FADs, refers to a family of genes that encode enzymes involved in the biosynthesis of polyunsaturated fatty acids, or PUFAs. These enzymes are crucial for the conversion of dietary fats into various important compounds in the body. Just like LP, almost all inhabitants of Europe carried the ancestral allele until the derived allele was introduced around 8,500 years ago by early Neolithic farming populations, reflecting adaptation to agriculture. Fads encode proteins which catalyze key steps in lipid biosynthesis pathways. These pathways synthesize long-chain PUFAs necessary for cells, particularly neuronal membrane development from short-chain PUFAs. PUFAs are essential components of cell membranes in the brain. They help maintain the fluidity and integrity of neuronal membranes, which is crucial for synaptic transmission and signal processing. They play a role in regulating neurotransmitter functions in the brain. For example, they can influence the production and signaling of neurotransmitters like serotonin and dopamine, which are involved in mood regulation, attention, and memory. Omega-3 PUFAs have anti-inflammatory effects. Also, they can help regulate the immune system by influencing the production of inflammatory mediators, which can be beneficial in managing chronic inflammation and immune-related conditions. In addition, they play a role in immune cell signaling and function. PUFAs can impact the activation, proliferation, and function of various immune cells, helping to mount an effective immune response when needed. Adaptation to fads was possibly crucial to various physiological processes, including inflammation, immune response, and neuronal function. These factors made these genetic adaptations important for conditions such as inflammation and cardiovascular problems, possibly improving fitness. And here is amylase. Another important adaptation to the agricultural diets of the Neolithic was the adaptation to starch-rich diets with amylase, an enzyme that plays a crucial role in the digestion of carbohydrates. As amylase is important in starch hydrolysis, the process in which large starch molecules react with water and break down into smaller sugar molecules, it is important to groups in arid regions which consume many starches, in contrast to people in rainforest environments or pastoralist groups, for instance. There are two types of amylase enzymes, salivary amylase, AMY1, and pancreatic amylase. Higher AMY1 copy numbers and protein levels likely improve the digestion of starchy foods and may buffer against the fitness-reducing effects of intestinal disease, possibly making AMY1 especially important to people in arid regions. Starchy foods often contain essential nutrients like vitamins, such as vitamin B. B vitamins are vital for converting food into energy, and they help metabolize carbohydrate, fats, and proteins, providing the body with the energy it needs to function properly. These vitamins help break down homocysteine. Higher homocysteine levels are associated with an increased risk of heart disease, showing how vitamin B supports heart health. They are also crucial for the production of red blood cells, helping prevent certain types of anemia by supporting the formation of healthy red blood cells. Overall, B vitamins are crucial for numerous bodily functions, including energy production, nervous system health, red blood cell formation, and more. Starchy foods also often contain minerals like iron, magnesium, and potassium. These minerals are important for healthy blood, regulating nerve and muscle function, proper development of bones, and a healthy heart. Deficiency in these minerals can lead to fatigue, weakened bones, and impaired cognitive functions. Maintaining adequate levels of these minerals is crucial for overall health. Deficiencies can lead to various health issues. And here is the adaptation to alcohol. When alcoholic beverages are consumed, the body metabolizes alcohol into acetaldehyde through the family of alcohol dehydrogenase, ADH enzymes. ALDH2 or aldehyde dehydrogenase 2 is an enzyme involved in the metabolism of acetaldehyde and other acetaldehydes within the body. Its primary function is to break down acetaldehyde, a toxic byproduct of alcohol metabolism, into acetic acid. When acetaldehyde accumulates in the body, it can cause damage to tissues and organs. This toxicity can result to a range of adverse effects, including nausea, vomiting, headaches, and rapid heartbeat. Acetaldehyde can also cause damage to DNA, potentially leading to somatic mutations. This DNA damage can interfere with normal cell function and potentially contribute to the development of cancer. Additionally, acetaldehyde contributes to oxidative stress in the body by promoting the formation of free radicals. These reactive molecules can damage cells and contribute to various health issues, including inflammation and chronic diseases. ALDH2 plays a crucial role in detoxifying the body from acetaldehyde produced not only from alcohol, but also from certain foods like fermented foods, baked foods, and fruits, environmental pollutants, and other substances. The ALDH2 GLU504 list allele has a high frequency only in East Asian populations with a frequency of between 30 to 50%. It's thought that the ALDH2 mutation spread from populations that occupied central China throughout East Asian populations. The ADH1B allele has also been under selection in populations in East Asia and elsewhere, as other populations have also developed alcoholic drinks and may have experienced similar selection pressures. The ADH1B enzyme converts ethanol into acetaldehyde. The introduction of fermented beverages or certain crops might have influenced the prevalence of genetic variations related to ALDH2. Some research suggests that the genetic variations affecting ALDH2 might have conferred certain advantages or disadvantages in metabolizing alcohol or acetaldehyde-containing foods, influencing the population's ability to tolerate these dietary changes. And so here's a recap of the significant adaptations. We have lactase persistence, fatty acid desaturates, amylase, and ALDH2 and ADH1B. In conclusion, the Neolithic opened up new food sources and lifestyle to populations with the domestications of plants and animals. With these changes, there are also some changes in the human genome to adapt to the new agriculture-based diets, genetic changes that stem from niche constructions like the adaptation of LP and adaptations to new types of foods such as starchy foods where amylase helped humans digest them without the harmful side effects. Selection of fads was important to various physiological processes, including inflammation, immune response, and neural function. Selection on ALDH2 and ADH1B in East Asian populations discouraged the consumption of alcohol. All these adaptations potentially increase the fitness in the people of the Neolithic and even the people of the present. However, there are still many open-ended questions in the topic of genetic adaptations during the Neolithic that should be future pursuits in this area. Nonetheless, the Neolithic was a landmark for recent human evolution because it resulted in many adaptations.